Host microbe interactions number 6. We're on slide 36. Now probably isn't going to be out of 48. We'll see. But at least this is slide 36. We are now looking at section 16.8 on page 427, damage to the host. We'll look at the difference between direct and indirect effects. We'll look at some general terms, and we'll look at 3 classes of exotoxins. We'll look at what an exotoxin is to begin with. So some damage that a bacteria does to the host is direct. The bacteria makes a toxin, the toxin is released, and that toxin affects cells, maybe it kills them. But as I mentioned before, a lot of the side effects we associate with diseases are actually our own immune response, particularly inflammation. So we can have direct effects of the toxin or indirect effects of it's not the bacteria's fault, it's our own immune response. And also the idea that this damage is to allow the pathogen to spread. Not quite that simple, but it's certainly an idea we can move forward with. Vibrio cholerae produces cholera, watery diarrhea, up to 20 litres a day. The bacteria is released out of the body, contaminates water supplies, and then contaminated water is drunk by the next set of people, and they get infected, and kind of the infection just snowballs. Bordetella pertussis, this is whooping cough, severe coughing, and pathogens are released into the air. This is a freeze frame of somebody sneezing. You can see the particles. This is the idea behind one idea behind the 6-foot social distancing, is that if you do cough and sneeze, a lot of the larger particles are going to fall out of the air within a few feet of you, although small particles can travel much greater distances. On to slide 38, exotoxins. Exo means outside or external. These are toxins that are made by the bacteria and then are typically released into the host. They are typically proteins. So proteins are one of the most, proteins are immunogenic, which means they stimulate an immune response. So we can produce an immune response, including antibodies, against just the toxins themselves. If you think back to the outcomes of antibodies, one thing here, neutralization, the surrounding of a toxin by antibodies neutralizes the effect of that toxin. If lots of bacteria all build up and they all release nasty toxins very rapidly, then it may be enough to overwhelm the immune response, overwhelm our antibody resources, and that's why diseases can kill us. If a disease is too quick, because we talked about the idea of the immune response building over weeks, several days up to weeks to generate antibodies. Vaccines, that's how we deal with things that are often so quick they kill us. We'll look at vaccines in more detail later on. If you do get a large kind of toxic influx into your body, we can use antitoxins. A rattlesnake bite, for example, you get an antivenom. That is a suspension of antibodies. Often antibodies grown in horses, and horses are picked because they're just very big and contain lots of blood. So they make lots of antibodies, and then you purify the antibodies out to a certain extent, inject them into the person, and they can be like a quick mechanism to neutralize toxins. There's very different ways we can group toxins. We can group them upon their function. Neurotoxins damage the nervous system. Enterotoxins damage the intestines. Entero means of the intestines, or it means inside. Cytotoxins damage a variety of cells. Cyto just means cell. So a cytotoxin just means cell toxin. And we've looked at things like cytotoxic T cells. The reason why they're called cytotoxic T cells is because they do indeed produce toxins that target other cells. But bacteria can produce cytotoxins as well. So there's going to look at 3 general types of exotoxins, and those types are based upon the structure and the mechanism. So number 1 in our list on slide 39. Our first kind of exotoxin is an AB toxin. And AB toxins are on page 429. It's called an AB toxin because it has these 2 parts, the A part and the B part. The A part is the active part, that is the toxic part. The B part binds. So B binds, A is actively toxic. This B region of our AB toxin can be highly specific. There's an idea, or indeed there are ideas that if we could pull off the A part and attach our own molecules onto the B part, different AB toxins will target different parts of the body, because once again it's a shape thing. There are receptors, or labeled binding sites, on the surface of cells, and our B part of the toxin fits into those binding sites. And different cell types in different tissues around the body are going to have different binding sites. So a respiratory infection that's going to produce an AB toxin has the B part that sticks into the respiratory binding sites on respiratory cells, and an entero bacteria that is going to infect the intestines will have a different B unit, because it's going to fit into the B. It's going to fit into the binding site that's on the surface of intestinal cells. So if we could harness the power of these binding units and their specificity, we could produce highly targeted drugs. That's the idea. So an example of an AB toxin is diphtheria. Diphtheria grows in the throat, releases an AB toxin that kills cells. The dead cells form this thing called a pseudomembrane, which causes many of the symptoms of diphtheria. It can block airways. In addition to that, the toxin can be absorbed and carried around the body to the heart and nervous system and cause additional damage there. One thing that this pseudomembrane does is it causes coughs, particularly coughing, and in addition to that, sneezing. Our diphtheria bacteria is here in the mouth, so coughing and sneezing are going to be one important mechanism to expel the bacteria out of one host into another host. This is one of those bacteria where it's AB toxin, the toxin that kills cells, was given to the bacteria from a bacteriophage. The presence of the AB toxin is a virulence factor for the diphtheria bacteria. And this kind of toxin on slide 41 are membrane damaging toxins. And these do just what they sound like they're going to do. These are toxins that damage plasma membranes. And in animal cells like humans, the plasma membrane is all we have. We don't have any additional structures on the outside, no cell walls in animal cells. So if you disrupt the plasma membrane, then you burst the cell, you lyse the cell, you break it open. These are cytotoxins because they are toxic to a number of different cell types. Cyto, cell, toxin, toxin. Hemolysins are membrane damaging toxins that lyse red blood cells. Pore forming toxins do exactly that. They insert into membranes and form pores. Streptolysin O from strep pyogenes. If we go back a few slides, number 27, we saw our S pyogenes making streptolysin O and sending it against phagocytes to kill them before they can arrive. Phospholipases, enzymes that break up phospholipids. And our membranes are made up of phospholipid bilayers. They break them down, and some very nasty diseases like gangrene here, caused by Clostridium perfringens. It's typically an enteric pathogen. That means that it is in the intestines, but if it gets into infected wounds, this is one example of the bacteria being in the wrong place. It will produce a toxin, and the outcome in this infection is called gas gangrene. Gangrene is a general term that relates to tissue death caused by lack of blood. The gas part comes from the gases that are produced during decomposition. So basically, the body is rotting in place. Okay, number 3 on our list of exotoxins are super antigens. So to understand the effect of super antigens, you're going to have to recall several elements from our look at the immune response. First of all, you need to recall MHC, major histocompatibility complexes. So MHC molecules, in this case MHC class 2 molecules, that is the way that certain cells talk to each other. We have here an antigen presenting cell. So a good example of an antigen presenting cell would be a dendritic cell. And we have our dendritic cell presenting pieces of antigen. Maybe it found those pieces of antigen in some tissue somewhere. There was a splinter and the splinter activated all of those reactions, causing inflammation. Dendritic cells uptook some of the microbes at the scene. They took them into their cells, they chopped them up into little pieces, and then they are displaying all of that microbes stuff, or at least their proteins, the bacterial proteins on their cell surfaces. And they're going to take those bacterial proteins and they're going to show them to T cells, in particular helper T cells. And the helper T cell will have been trained to recognize harmful or non-harmful, or self and non-self. So here we have an antigen-presenting cell presenting an antigen, the red blob. It's a peptide, it's a piece of a protein, and in this case we have the T cell receptor, and the T cell is stimulated by the antigen because this is an antigen that the immune system has seen before, and it's an antigen that has been categorized as being harmful. So the antigen-presenting cell says, look at this, what do you think? The T cell says, yes, I recognize that, that's harmful, and it's going to release cytokines. It's going to trigger an immune response. Sometimes the antigens that are presented by antigen-presenting cells are not harmful. In this case, this kind of yellow diamond is presented to the T cell. And the T cell says, no, that's nothing to worry about. T cells need to discriminate because of the damaging effects of activation. When the T cell releases these cytokines, it's going to have damaging effects to the host. So if the T cell doesn't recognize an antigen as being harmful, it will not initiate an immune response. However, in the presence of a super antigen, anything presented by an antigen presenting cell will be seen as dangerous. This molecule that was previously not a problem for the T cell is now a cause for the T cell to activate and trigger an immune response. In many ways, the super antigen, this purple blob, is short-circuiting this system. So no matter what the antigen-presenting cell, no matter what the dendritic cell is showing, the T cell is always going to activate. And that means lots of activation, lots of cytokines, lots of inflammation, and lots of deleterious effects on the body. So, MHC class 2 presents exogenous antigens, antigens from outside of cells, from external sources. Dendritic cells and macrophages can present using MHC class 2s. They'll present to T cells. The super antigen will short circuit. The T cell will activate no matter what. There'll be massive cytokine release, typically excessive or uncontrolled release of inflammatory cytokines, and systemic inflammation is extremely serious. Toxic shock syndrome toxin is a type of superantigen that will trigger this massive cytokine release and this systemic, potentially fatal inflammation. Staph aureus, and once again our good friend strep pyogenes. You may have heard this in relation to tampons, particularly super absorbent tampons. The kind of the nasty, well, Upshot is that the blood that accumulates in the tampons is a great culture media for bacteria, such as S. aureus. They grow in the tampon and then they release their toxin. The toxin then causes a massive systemic, often called a cytokine storm, just because of the severity and size of the cytokine release. Okay, study guide for this section. Damage to the host, exotoxins, know how they work and some examples of diseases they contribute to. Know that idea of the different effects of neurotoxins and enterotoxins and cytotoxins. So they're a group based on function. Be able to distinguish between direct and indirect, and then know how these types of toxins work. Identify an AB toxin versus a membrane damaging toxin versus a super antigen. I've got direct damage down here. And then for each one of these, know how they work and what kind of diseases they cause.